Good morning once again, and welcome back to this course in Fundamental Principles in Catholic Bioethics. And we begin, as always, with a little prayer in the name of the Father, the Son, and the Holy Spirit. Blessed Trinity, we love you, we adore you, and we thank you for the many gifts that you give us on a daily basis, the gift of life, and again for having us here today. Bless our gathering, inspire us to go deeply into our nature, the nature that you have given us according to your image, that also has a biological beginning according to the science, and that teaches us that we descend from common ancestors. We ask that you inspire us and that you bless all our gatherings, bless all our doings, according to your divine will. And we continue to pray for those who are in most need of your divine mercy at this time. In the name of Christ our Lord. Amen. So today we're going to go into the forces that move evolution, that cause evolution, that may cause evolution, okay? And just to remember that we are dealing in a probabilistic model. Maybe it's a little too bright. Is the lighting okay, or should I turn on some lights? Better? More? Better? Or no? You're okay? Yeah? All right. Okay, just to make sure folks online should be able to hear me. Yeah.
Yep, I'm good.
Okay, thanks. Right, so keep in mind that we're moving in this probabilistic world, okay, or worldview. But the probabilistic worldview is statistical. Chances are that this will happen, and even you can put a percentage on it. However, we live in real life in a deterministic world, meaning that we really exist here and now, physically. And so that's always a challenge to translate from the probabilistic world view to the deterministic worldview, okay? But in evolution, because it has happened already, all right, what we see is the result. But the process itself has already happened. And so we infer, like Meyer says, we infer what has happened, and these are the main causes that could cause evolution, the main forces or pressures, if you will, which are, in brief, mutation, migration, drift, and selection. We're actually going to cover the first three today and leave selection for the next lecture, which will involve natural selection, but also sexual selection, and an example that we mimic nature by artificial selection. All right? Okay, so any questions or comments? No? Going forward from last time. Let's look at first a mutation. I give you a little outline that you can follow. So in mutations, we're talking about, so let me say what level also these are occurring. Mutation is going to be occurring at the level of the DNA, of the genetic code of the chromosomes or chromatin. At that level, it's at the molecular level, okay? Migration and drift are going to occur at the individual/population level. All right, because no individual exists alone in real life. In reality, every individual is part of a population, even if that population is just 2 or 3, or it could be a huge population of millions, like bacteria on a single colony. All right, but when we talk about individual species, individuals within nature, it's always in the context of some kind of population. So migration and drift occur at that level, at the level of population, and selection also at the level of population. But eventually it has to go to the individuals within that population, because different individuals within a particular population will be affected differently. depending on many different circumstances, depending on the variety, on how fit they are to survive the particular environmental stress that is happening. Okay, so you see the different levels. Mutation is at the molecular level, whereas the other three are at the organismal / population level, the whole organism. All right, so let's get right into mutation, which actually covers three aspects. The change in the DNA sequence, the change in the DNA sequence could be either by actual mutations, different types of mutations that we'll talk about in a minute, or it could be by a process called recombination, which is also known as crossing over, and that occurs during the formation of the gametes. What are the gametes? The gametes are the sex cells, right? Egg or sperm, which is the package that transmits the inheritance from one generation to the next of each particular species. And then the third process is called transposons, which is basically segments of DNA that jump from one place to another within the same cell. All right, so just to refresh the mind, if we start with the DNA sequence, the actual DNA sequence is called the primary level of the structure of DNA. This is the actual sequence. These are called bases, right? A, T, G, C, and they're chemical compounds, and they're all paired up. So I tell you A pairs with T and C pairs with G, right? The straight lines with straight line and the curved lines with curved lines. So each one of these paired up is called a base pair. So AT is a base pair, GC is a base pair, etc. These are base pairs here, right? And they're complementary base pairs. If I give you one sequence, you are able to define for the other sequence because, again, C and G always pair up and A and T always pair up, right? Okay, so this is called a complementary base pairing of DNA at the primary level. That in nature doesn't occur just flat like that in a two-dimension form. See that this is expressed in a two-dimension shape here, right? Just flat on the screen. But that's not how it occurs in nature. It turns out that this ladder, this DNA ladder, is twisted into what is known as a double helix. So here's a double helix because there's one helix and the other one intertwines around it. So it's a double helix, right? This is what we call the double helix, which is a stable DNA. And then this double helix also coils. Remember around the little, what are these histones? What are these? They're proteins, little protein balls that are paired up also. There are 4 pairs, right? That's too much detail, but anyway, these little balls of proteins are called histones. The DNA winds around it almost 2 times. And this is known as the basic unit of chromatin called the nucleosome, the little body inside the nucleus, and it's a basic unit of functional chromatin. And then the nucleosomes also coil on top of each other. So you got coil on top of coil on top of coil is what we call supercoil, right? Eventually, the whole fiber within each nucleus of each cell of each living organism, whether it's a plant, a fungus, or an animal, is going to form the chromatin, which is the functional DNA in a cell when the cell is not dividing. And then when the cell is dividing, the metabolism is brought down to a minimum because the cell is now going to divide, typically split from 1 into 2. So that chromatin condenses even further into little tiny visible bodies that we call chromosomes, right? Distinct chromosomes. And each species has a distinct number of chromosomes. All right, for the human is how many? 46 chromosomes, and they're always paired up, so it's actually 23 pair. And in a strict sense, the 23 pairs only exist in whom? In a male or female? If we talk about actual 23 pairs, identical pairs to each other, only in the female because they have to, the sex chromosome has to be XX to be identical. If it's XY, then it's not actually identical, right? It's almost identical. All right. Okay, so you're all clear on the structure of DNA at the different levels. Primary, secondary, tertiary, and the quaternary is actually the functional structure. So all of these occur at the level of the DNA molecule. And let's see how that is so. All right, the DNA molecule will be affected either in sexual reproduction or in asexual reproduction. All right? So mutations, transposons, and recombination are going to affect the DNA of key cells. Of course, we can have mutations in our body. But if the cells in our body mutate, will that be passed on to the next generation? No, only if what cells in our body mutate. Only if the gametes in our body mutate and those gametes get to be passed on. All right, then we have a passing on of the mutation. But I can have all the cells in my body mutate. If my sperm is not mutated, I'm the only one who has become a mutant. But my progeny will not be a mutant unless the actual DNA in the sperm that gets to fertilize another egg, only then will that mutation be passed on, see? So a lot of filters here that are set up by nature. All right, so we need to look at sexual reproduction, and we also need to look at asexual reproduction because a number of organisms also reproduce asexual. Generally, the simpler organisms and the smaller organisms reproduce asexually. All right, so let's look at what I mean by the mutations, transposons, and recombination at the gamete precursors. The gamete precursors doing a process called meiosis, which meiosis is kind of a double mitosis. Mitosis is cell division, okay? All right, so let's look briefly at sexual reproduction, just in very general terms, and we'll cover it in greater detail in the course in the spring on the beginning of life bioethics. But for now, we have the female reproductive organs and the male reproductive organs. I'm focusing on the human for now. You're familiar with these diagrams, I'm sure. Just to review, the cervix is a circular muscle. These are all, this is muscular tissue, mostly on the female reproductive structure. It has muscular tissue, which are smooth muscle, meaning that they're not directly connected to any bones. All right, and they're also involuntary muscles. In other words, we don't have direct control over those muscles, voluntary control. The muscles over which we have voluntary control are called skeletal muscles, which are the muscles connected to bones, right? And I can say to myself, move my arms because I have muscles that are connected to the bones of the arms. But I cannot tell my stomach voluntarily to squeeze, all right, because those are involuntary muscles and they're connected to a different part of the nervous system. At any rate, a very important muscle is the cervix, which is a circular muscle at the entrance of the uterus here, which separates the uterus from the vagina. The vagina is actually the opening to the exterior, to the environment, and therefore subject to pathogens and foreign objects. So the cervix generally is closed tight and maintaining integrity so that the body is not compromised, because this is a direct opening into the internal organ of the female body. The uterus, the normal function of the uterus is to receive an early embryo that will implant. The uterus becomes spongy and thickened to receive that embryo and then to nourish it, in the case of the human, for 9 months before birth. Then these tubes, which are known as the fallopian tubes, again, they are smooth muscle, and eventually they branch out into a little hand-like structure, which is called the fimbri, I drew a blank, but it's like a little hand that covers over the ovaries, but it's not directly connected to the ovaries. Okay, the ovaries are the actual sexual glands, the primary glands, and the ovaries are the ones that contain the eggs. In the male reproductive structure, the main gland is the testicle, which has a series of tubes on top of it called the epididymis. And the epididymis holds the sperm for maturation, for further maturation. And all that is contained within a sac of skin that is called the scrotum. And you notice that the testicle and the ovary are analogous structures, analogous meaning that they have the same or similar function, right? They produce what? The gametes, they produce the sex cells. They produce the sex cells. So the ovaries produce eggs and testicles produce sperm. And therefore, these glands, the ovary and the testicles, are analogous in that they have a same or similar function, namely to produce the gametes. And the production of gametes is called gametogenesis. However, in the body, physically within the body of the woman or the man, they are, even though they are generally in the same area around the pelvis, they're in the pelvic region, they are locally in very different parts of the body. All right, namely one is very internal and the other one is actually external. If you notice, the ovaries in the woman are basically as far in as you can get into the body. All right, they're within the pelvic region, and because that's for protection, because it has all of the body for protection, right? So the reproductive organs of the female are very well protected within the body itself. Whereas the testicles actually hang out of the body of the man into this scrotum bag of skin. And you would think that's a dangerous situation, right? Because here is essentially what contains the inheritance of the next generation. And they are exposed to the environment. And that is for a particular reason, and that's why I have this little diagram here of, it's actually an electron photomicrograph of an egg, a human egg, and a human sperm to scale. So you can see that the human egg is much larger than the human sperm, right? These are to scale. It's an actual electron photomicrograph, including this little layer, which is probably zona pellucidea, a layer of cells that protects the egg. The point here is that the nucleus of the egg, of the sperm, is called the pronucleus, has how many chromosomes in the case of the human? Only 23. They're not paired, right? Only one set, 23. How about the nucleus of the egg? Also 23. Unpaired, because the pairing is about to occur. But so it's also a pronucleus, a female pronucleus and a male pronucleus. So the nucleus here is also about the same size as the sperm nucleus. Because it's a pronucleus, it has the 23 chromosomes on each. But look at the size of the egg. So what's the big difference in volume here? If the two pronuclei are about the same size for the egg and the sperm, what is drastically different in volume for the sperm and the egg? That is not the nucleus, it's within the cell. It's a major component of the cell. Is that environment, that soupy environment where metabolism occurs. We call it cytoplasm. Cyto, reference to the cell. Plasm is a soupy environment, all right? Gelatinous type. Bottom line, the egg has much more cytoplasm than the sperm, which allows it to live longer, for sure. There's more nutrition here. Whereas the sperm is just basically that package of a pronucleus. Oh, okay. Which means that the sperm is much more susceptible, especially to temperature, all right? And what happens at the body temperature, what's the body temperature? In Fahrenheit, which is simpler for us. 98.6, right? The normal body temperature, 98.6 in Fahrenheit, which, by the way, that translates into what in centigrades? About 37 centigrade, all right? Let's stay with Fahrenheit, which is a temperature that we're accustomed to. What's temperature in here right now, approximately? It's on the cool side, right? Not as cold as other times when it's been like in the 50s. Okay. Maybe, what would you say? Is this in the 70s? It's in the 70s, probably. Maybe low 70, right? Maybe 68, 69, getting into the 60s, right? Yeah, not as bad as the 50s. Okay, let's say it's in the high 60s, low 70s, right? Imagine if we raise it up now and we can go into the 80s. It's going to get warm, right? What if we go into the 90s? Probably in the 80s outside. Probably get into the 90s by the midday. In the 90s, it's pretty hot, right? 90-degree weather, about 95. 95 is pretty hot, right? Fahrenheit. Well, we're still 1 degree off, almost 2 degrees. 96 degrees Fahrenheit inside our body right now. like the heart, the lungs, all that, the intestines, all that area, the viscera, that's all 96 degrees. Okay, 98 degrees Fahrenheit. That's very warm. Too warm for the sperm. And so the sperm at that temperature actually die, or they can get mutations, because one of the causes of mutations is heat. So that's why they have to hang outside the body, right? They have to hang outside the body because otherwise the body temperature kills them. And in fact, I'll go a little further in detail, the scrotum actually moves up and down the body. And when it's very hot, the scrotum hangs down. Like athletes, if they're exercising, the scrotum hangs down to get those testicles as far away from the body as possible. All right, and to cool the testicles and the epididymis. Where it's very cold, that scrotum crunches up close to the body to get some body warmth. And that's a natural process that is involuntary, it's temperature related. Okay, so very different. The eggs can actually tolerate that warmth because of the amount of cytoplasm it has, but the sperm is much more delicate. And that's maybe another reason why also it takes millions of sperm to have a functional ejaculate, a fertile, potent ejaculate, has to have on the average of 100 million sperm. And it only takes 1 sperm to fertilize, right? Because many of them can be damaged and so forth. Okay, so we're clear. Gametes are the sex cells, and they're either egg for the female, sperm for the male. That's standard with not only the human, mammals, and many other species of plants, animals and plants that reproduce sexually. They follow the same process. Of course, they don't have the sexual organs. See, what is the sexual organ of the plants? In flowering plants, it's going to be the actual flower, all right? And in non-flowering plants, like pine trees, it's going to be the cone. And there are male cones and female cones. All right. So again, focusing on the gametes, they are produced by a process called gametogenesis. By this point, you should know genesis, beginning or origin, and the process is called gametogenesis. So the gametogenesis of sperm occurs within the testicle. This is a diagram of a cross-section of the testicles. In these little tubules, these are the beginning of the tubules. They're called seminiferous tubules. Seminiferous tubules. And they are the ones that produce sperm. So the production of sperm is called spermatogenesis. Spermatogenesis. The production of egg is going to be called oogenesis. Oogenesis. All right, so sperm is produced in here, and this is the critical area then where mutations may occur. Mutations may occur by either ultraviolet radiation or by chemicals or by heat that can cause mutations during gametogenesis, all right, during spermatogenesis. Once the sperm is generated here, it will move with the fluid, which becomes part of the semen, will move through these tubules into other sets of tubules at the top part of the testicle, beginning to form what is called the epididymis. And these tubules continue to connect and consolidate into a larger tubule. that is the main body of the epididymos as such. And eventually, when that tubule leaves the region of the scrotum and goes back into the body, it goes through the tubule is called the vas deferens, and then it picks up more fluid, semen from the prostate gland and so forth. But this is where the sperm are stored and continue their maturation process. For the egg, is a little different. This is a model of an ovary that has been sliced in half and opened, right, like that, fan shape. And so we see, starting with the little tiny eggs, oocytes, they're called oocytes. Cyte is a reference to cell, O or egg. And the egg will eventually mature into what is known as a follicle. And when the follicle continues to mature, the follicle will rupture and the egg will be exuded, will come out of the ovary proper and into the infundibulum. Infundibulum is the little hand shape, and move a little bit up to what is known as the horn of the tube, okay, or the ampulla here, and will stay there for about 24 hours awaiting fertilization. That's at the maturation of an egg.
Father, you said that it's not behind this colon and it's not connected to the ovary.
Exactly. It's adjacent, it's overlapping the ovary. Okay, so the ampulla, the infundibulum is a little like a glove, all right, that is overlapping the ovary, but it's not a single piece, it's not a single structure, there are two. And that slides and actually moves back and forth. That's what causes some of the pain at ovulation, is the movement of the hand back and forth to wrap around so that it actually captures. It's like a miniature baseball game going on there. Yeah. The follicle ruptures, the egg is released, and then the infundibulum catches that egg. All right, it's in very close proximity and it's all filled with fluid, of course. That's also why occasionally one of those eggs can slip out into the abdominal region. And if the egg was fertilized, you actually get the growth of an embryo in the abdominal region, extrauterine. Extremely dangerous, but sometimes it's happened. And sometimes those babies have actually been taken to term. We'll talk about that next semester, but very interesting. And anyway, what I wanted to point out here, once the follicle ruptures and the mature egg is released, then what is left of the follicle becomes this other little body called the corpus luteum. And it has a function because it continues to release hormones that will maintain the ovary thickened and preparing for a potential implantation, because an ovulation has occurred. You know, a mature egg has been released, so the whole body gears toward a potential implantation, a potential pregnancy, all right? It will affect then uterus and also affect the breast for getting ready potentially for maturation. Now, what I want to say is that the analogy between ovary and testes is at the general level of producing gametes, whether they are egg or sperm, right? But in specific, there are differences, and again, I'll go into more detail next semester, but essentially, a woman is born with a set number of eggs, all right, which are premature, they are in arrested development, and it's in the millions. So a baby girl in her ovaries, in her little ovaries, she will have millions of immature eggs that are arrested. And they're arrested for about a dozen years until menarche, her first period, which means that she has matured the first egg. And from then on, she will continue to mature, on average, one egg every month, a lunar month of 28 days, and so forth, for all her fertile years, which are approximately 3 dozen years until the 40, more or less. I'm giving you general numbers, all right? So a woman is born with a set number of eggs, which are in the millions originally. By the time she hits menarche, when she's anywhere from 8 to 12, all right, on average 10 years of age, she's down to thousands, hundreds of thousands of eggs, but no longer millions. Most of those eggs little premature eggs have degenerated and have been reabsorbed by the tissue of the ovary itself, right? But of course, she only needs, we can do the calculation, no, I'm not going to take time to do the calculation, but she only needs a few hundred to be fertile for her whole 30 years of lifespan, because it's only one per month. At any rate, what I'm trying to point out here is that the baby girl is born with a set number of eggs for life, and the vast majority of those become degenerate and are reabsorbed by the ovary tissue itself. But she only needs a very few to be fertile all her years of fertility. In contrast, with the man, the baby boy is born with testicles normally, all right, but they are immature, and it also takes, on average, about a dozen years, anywhere from 8 to 12 years, when that young man will finally produce the first fertile semen with sperm, which will be in the millions. But in contrast with the female, the male is not born with a set number of sperm, of premature sperm, okay? Rather, they are precursor cells that are found within the tissue of the seminiferous tubules on the inside, on the inner lining of the tissue, and those precursor tissues are the ones that make the sperm, all right? And that, again, takes about a dozen years to mature. And then from that point forward, once the testicles are mature, every day, every hour of every day, until that man dies, those testicles, on average, normally are continue to produce sperm, all right, which are moving through the vessels into the epididymis and then stored there until an ejaculation occurs, which will then move the sperm out with the semen and the fluid of the semen out through the vas deferens and the other structures of the body. What happens to all that sperm? It's millions of sperms that are being produced every hour of every day for dozens of years. We don't just blow up, right? The vast majority of that sperm becomes old within a few days and is reabsorbed by the tissue itself, also by the epididymis, okay, and the vas deferens, mostly the epididymis, the inner lining of the epididymis reabsorbs the old sperm. So the sperm is being refreshed periodically, naturally, even without ejaculation. Of course, ejaculation is a dramatic event that drains a lot of the sperm to the tunes of millions of them, and then is replenished slowly but surely within a few days, and sometimes just a few hours, depending. But you see the difference, okay? Whereas a woman is born with a set number of eggs, the man is not born with a set number of sperm, but rather the sperm are produced when the testicles finally mature. Okay, enough detail there. Let's move forward. So you see, when if mutations are going to occur, mutations that will affect evolution, in other words, that will affect the next generation, the mutation has to occur at this level, at the level of the oocytes or at the level of the sperm. Okay, if any other part of the body, any other tissue, organ of the body is mutated and doesn't affect directly either the ovaries or the testicles, then that individual has become a mutant, but it's not necessarily passed on to the next generation. All right? Okay.
What are some of the mutation examples that can occur?
Exactly. I'm going to get into that detail in a few more slides. Okay, so we're going to look at that. So this is a general picture, just to know then that the testicles and the ovaries are the ones that would be affected by either radiation or chemicals or heat to cause mutation, all right? And that's why they're very delicate organs, because if mutation occurs there, then there's a possibility that that mutation may be passed on to the next generation. There still has to happen. What needs to happen? Well, the one, let's say that the sperm in here actually mutated, all right? That sperm still needs to go through all this process and come out this way and actually fertilize an egg from here, all right? And the chances of that happening are astronomically low, but it does happen because we're all here today, okay? But you see, so there's an excess of safety mechanisms, if you will, just by the sheer number of excess of these cells, which Darwin noted, by the way, and we're going to get into that in the next lecture, this excess that one finds in nature. All right, so let's go, let's now look at asexual reproduction, where, so this is where the mutation would occur to make it, to have an evolutionary effect, right? Follow? Okay, so let's look at asexual reproduction. Asexual, the A prefix negates the next, so it means non-sexual reproduction, right? But it's still reproduction, meaning that the genetic material is somehow passed on to the next generation. So the fact that it's asexual reproduction does not mean that there's no reproduction. It is a reproduction, in other words. And at the molecular level, reproduction means passing on the genetic package to the next generation of cells or organisms, whatever they are. All right, on the one hand, we can look at bacteria, which are what? Prokaryotes, so they don't have an organized nucleus. And this may hold true also for archaea, which in this sense are similar. What happens in them is mostly what is known as lateral gene transfer. Lateral gene transfer. Okay? And it goes this way. Here's a bacterium, and here's another bacterium. All right? One bacterium, several bacteria. So this is one bacterium, this is another bacterium. Between the two of them, these are two bacteria. Right? So the normal structure, there will be a chromosome, which is typically circular on both, but also another little circle of DNA, which is an independent circle of DNA that is called a plasmid, all right? And that circle of DNA is independent of the main DNA, the main chromosomal DNA. All right, so, but it's also a little circle of DNA. And then bacteria also have these little tubes that come out. They're called pilus, one pilus, several pili. It's also a reference to a hair-like structure, all right? So we have the little hairs in our body are called pili on our body. So the pilus connects one bacterium to the other, and that pilus then brings the bacteria together, and a gap is generated between the two bacteria. Then proteins, protein structures transfer, essentially they transfer the plasmid from one bacterium into the other bacterium. And there's also, you can see there's a duplication effect here, which is called replication. The end result, this is in general terms, the end result is that the two bacteria now end up with the same plasmid in either side. Okay, so there's a transfer of DNA information from one bacteria to the next through the pilus by a process called lateral gene transfer. Okay. So technically it's considered a type of asexual reproduction. Other types of asexual reproduction are different types of clones. Now, the reference to clone is that the genetic material is the same. It's essentially identical, all right? And that's a reference to the clone. It's at the molecular level, the genetic material, the genetic information is essentially the same, One example in plants is called vegetative propagation. If you pull out a tuft of grass, you will see that many of those tufts have little shoots, little lateral shoots that go into the ground and generate another tuft of grass. But you notice that this little shoot, all right, connects tufts one next to the other, and they're all from the parental, the original tuft of grass. And so these are all clones. These are all clones. If you cut that shoot on both sides, this little tuft becomes independent and can become the parent of other little tufts through what is known as vegetative propagation. Vegetative propagation, because there's no sexual reproduction. Now, this occurs in many plants, especially grasses, But you will notice that grasses also have little flowers, all right? Small, tiny, very with drab colors, not very brilliant colors, at least to our eyes. They have flowers, which means that they can also reproduce sexually. But remember that sexual reproduction energetically involves at least twice the amount of energy. So it's easier to reproduce asexually, all right? Energetically wise, easier to reproduce asexually. But then you have essentially identical clones. So if there's a mutation on the parent, through the shoot, that mutation will be passed on to the progeny. Please? Yes.
This vegetative propagation would also refer to any plants that are tube-like, like grow on the ground.
Yes, yes, tubers also, and also
like yams and potatoes.
Yes, those are all tubers, right? It's vegetative when the little eyes come out and you cut out an eye and plant it. That's another type of vegetative propagation. Also for orchids and bromeliads at the base, they put out little babies, all right? You break them off and plant them. They also take off. Those are all general categories of vegetative propagation, right? Because there's no sexual reproduction occurring. There's no union of gametes. There's no fertilization. So that's why they're close. They have essentially, each one of those tufts, each one of the progeny, has essentially the same genetic information as the parent plant. And it's very common in plants. An alternative that happens in animals is called budding off, and it happens again in lower animals, simpler animals, coelenterates, for example, hydras, anemones, right? They grow a little bud on the side, and the bud continues to grow and eventually forms tentacles, which are more specialized tissues. And eventually that little bud breaks off into a new little hydra. And then when that hydra matures, it can also put off other buds, and that's called budding off. And it happens typically in simpler, lower animals that have a lot of regeneration tissue. But you see they're clones because this new little hydra, this progeny of the original hydra, has essentially the same DNA information in it, all right? The same DNA information. Another type is simply binary fission. So fusion is joining, fission is dividing, right? So nuclear fusion is the joining of nuclear particles. Nuclear fission is an atomic bomb, is the breaking down of the division, the breaking. So fission is division, and this type of fission, binary fission, because the cell splits into two, happens typically in protozoans. Remember the protozoans, protists, like amoeba, paramecium, and so forth, okay? These microscopic organisms that typically live in an aquatic environment, they can split in half, and the daughter cells would be essentially identical as the parent cell, meaning that the DNA inside the nucleus of each of these is going to contain essentially the same information. All right, so that's another type. Mitosis can also be seen as a type of binary fission, because in mitosis, one cell essentially splits into two identical daughter cells. So it's another type of asexual reproduction, if you will. Then there's another type that is called fragmentation and regeneration. This happens in other animals that can be higher order. For example, these are echinoderms. This is a starfish. And here you can see this starfish lost one of the 5 arms, and it's being regenerated. Okay. And the converse is, here's another starfish. The fallen arm, the broken off arm, actually regenerates into a new starfish. And the trick here is for the fallen arm, for the cutoff arm, as long as the cutoff arm has a little piece, at least a little piece, some tissue from what is known as the central disc. This is the central disc of the starfish, right? As long as it contains some tissue of the central disc, that central disc contains a lot of stem cells and has the capacity to regenerate a whole new starfish. Of course, it's not going to look like the parent starfish, okay, because time has elapsed, but it will regenerate to various degrees of size. But the point is that this is definitely a new individual that can function normally with a longer arm, and the other regenerated arms are a little smaller. Okay? Questions? No? You follow? Okay, so these are examples. We've seen sexual reproduction, we've seen asexual reproduction. Because why? Because we're talking about mutation. And again, in the sexual reproduction, the mutation to be passed on to cause evolution would have to be at the level of the sperm and egg in sexual reproduction. And in asexual reproduction, again, the mutation would have to be at the level of the DNA. So in the case of the bacteria and the archaea, it would have to be at the level of the plasmid here, or if a segment of the chromosome is going to be passed on at the level of that chromosome. And in here, again, the mutation would have to be at the level of the cells that are generating the new individual. Okay, the cells that are generating the new individuals need to be mutated for that mutation to pass on. If the mutation of this hydra only occurred on the tentacles, well, it's not the tentacles that is generating the new bud. So that mutation that only occurred on the tentacles is not going to get passed on. All right, so you really have to localize where the mutation has occurred. Same here, essentially, if the mutation occurred in the cytoplasm, it's unlikely, but on the nucleus, yes, because the nucleus will be split into two new nuclei. All right, now, types of mutation, to Andrea's question. We can talk about either point mutations or segment mutations. In other words, we can look at a single base pair change, whether it's a substitution, an addition, or a subtraction of a single base pair. Those are called point mutations. Or we can look at a segment of the DNA, several base pairs. It could be small or large, but all those may have an effect because they change the reading frame, they change the code. And the code is sequential. All right, so let's look at that into a little more detail here. Let's look first at point mutations. And the typical point mutations are either substitutions or deletions. All right, either a substitution or a deletion. Here's an example of a substitution. You see this mRNA, okay? The M stands for messenger RNA. This mRNA carries the code of a DNA, of one of the strands of a DNA. The DNA has the genetic information, right? So the DNA is precious. It's like your original copies, your originals of whatever document you have. It's a precious document. You keep that original document in a filing cabinet inside a room, inside an office on the lock. The cabinet is locked, the room on the office is locked, and the building where the office is locked. Right? So at least have 3 locks before you get into the actual original document. That's like the DNA inside the nucleus. You get the nucleus, get the DNA inside. Well, first there's the chromatin structure that is supercoiled, so the information is crunched in there. Then you get the nucleus enveloping that chromatin. Then you get the cell membrane enveloping the nucleus and so forth, right? So the DNA code itself always stays inside the nucleus. However, the proteins are produced outside the nucleus in the cytoplasm, in the soup, in the metabolic soup. And so that code has to be transferred somehow to the cytoplasm. And it's transferred by another molecule that is called the messenger RNA. It's RNA because it's a single strand instead of a double helix, instead of a double strand, all right? But it is it's the complement read of the DNA. Because again, we said that the bases match. So for example, this read, right? Starting with this first C, can you see it's a little small? Okay. On the messenger RNA, what's the first letter? What's the first base? C, exactly, which stands for cytosine, all right? And so what is the complement of this C in the DNA? Exactly, guanine G, right? So wherever there was a G, it became a C in the RNA. And that's how it works. So you can tell me the code here, the genetic code of this sequence in the DNA, in the original DNA strand. It is going to be G, G. Oh, now we have a U. Okay, I have to tell you that U replaces T. All right? U in RNA is the same as T in DNA. And what's the complement of T? A for adenine, right? So in the DNA sequence, this would be G, G, A, C, C,
And DNA T.
T, right? And so if I give you the mRNA, you can tell me what the DNA sequence is. All right, that's how it works. That's called fidelity. All right, it's faithful. All right, but the RNA is what is transferred out. It's the message. That is, so it's like a photocopy of your original document that leaves the filing cabinet, that leaves the office, that leaves the building, or maybe goes to another office within the same building, right? That's the messenger RNA. Then the messenger RNA in the cytoplasm is going to be transcribed into the actual protein. And that's what DNA codes for. DNA codes for proteins because the proteins are the ones that do the work in the body, all right? For example, enzymes are proteins. All right, and so the proteins are made up of little building blocks that are called amino acids, all right? There are 20 amino acids in nature, and that's part of the genetic code that we'll look at in more detail next semester. Basically, three bases in sequence code for one amino acid, 3 bases. So the first 3 bases are what? What are the first 3 bases on this mRNA? What are those first 3 bases? CCU are the first 3 bases, right? And so these 3 bases code for an amino acid called proline, which is represented by the first 3 letters. And so this little 3 bases is called a triplet, all right? And it's also called a codon, and it has a number. So we can put the number of codons here in sequence. And this particular codon has 146 triplets, all right? Too much detail, don't worry about it. I'm trying to get to the point that every 3 bases, there's going to be 1 amino acid. For this TRP is tryptophan, it's another amino acid. And what is the genetic code for tryptophan? What is the triplet for tryptophan? It's going to be U, G, G, all right? Which on the DNA is going to be A, C, C, right? Okay, and so on and so forth. There's another one. Now we get to this one. This is glutamine or glutamic acid is CAG. This is the one of interest because we're looking at a point mutation, all right? A single base pair substitution. All right, so on the codon number 39, the triplet base is on the mRNA is CAG, which means that on the DNA, C complements with
G.
With G, exactly, and A complements with T, and G complements with C, right? So we'll have G, T, C is the triplet base on the DNA. G, T, C, all right. That G on the DNA is going to mutate to an A. Instead of a G, is going to mutate to an A. The A is going to code for what in the mRNA? It would code for T, but the T is replaced by U, and there's the U. All right? So on the DNA, this G, on the DNA, this G, if that G mutates into an A, which is a very similar structure, all right? If that G mutates into an A on the DNA, then that A is going to be U in the mRNA. And here's the substitution, all right? Now it turns out that CAG triplet codes for glutamine, but UAG on the triplet codes for stop, to end the protein. So what happens? The protein ends prematurely. This is on codon 39, and the whole sequence is 146 codons. So 146 minus 39 is 130 something, right? So there's still more than 100 amino acids missing in this protein. It truncates it, all right? So it's only about 1/3 of the actual protein. What happens? What is this protein? Well, this protein is called beta globulin, all right, or beta globin. It's part of hemoglobin. It's part of the red blood cell. It's a critical protein in the RBCs, and it has to do with iron, and it has to do with respiration. The bottom line is that this type of mutation, a single point mutation on the DNA in this critical base, this critical letter, will cause a truncated beta globin on all the RBCs of the body and will lead to anemia, a weakness. So people that have this mutation can live but are weak, and it's called thalassemia, beta thalassemia. Remember I told you about the Chinese mutating, doing an experiment? That's what they corrected on the embryo. They corrected this letter, this mutation, they corrected it, but of course they did not implant the embryos, they just proved that it can be done. Then they discarded the embryos, right? They killed the embryos, but that's what they changed, that mistake. Okay, so this is an example of a point mutation. Let me turn it down a little bit.
Father, can I ask a question?
Yes, and it's a type of substitution, you see? Yes, go ahead.
Why does that happen? Why does the mutation happen in the first place? Like, why would the DNA make that mutation?
Exactly. We're going to get at the agents that mutate. I've been saying them just orally, but you'll see them graphically. They are radiation or chemicals, mutagens, you know, that carcinogens, for example, chemicals, or heat. Those are the three main sources of mutations, okay? Radiation,
thank you.
Chemicals, and or heat.
So if you get cancer while pregnant, skin cancer because of the sun that it mutated, then what happens to it?
Well, it depends. If it's skin cancer, then it's on the skin of the pregnant mom, right?
Okay, so it is.
But
It leukemia?
it depends on, exactly, those cells could be passed on to the baby. So it depends on what kind of radiation, what kind of chemicals. Some chemicals can go through the placenta into the mom, all right? Antibiotics, for example, and so forth. So in the developing embryo, those cells are very delicate, very pristine, and there are fewer cells, especially in the early embryo. So those are the dangers there. All right, so it really depends on the type of radiation, the dosage. It depends for X-rays, for example, because they penetrate through the whole body. It depends on the type of chemical, the dosage, and the type of heat also. Heat is a little less because obviously once we feel very hot, we get away from the heat, right? But the other two, chemicals and radiation, are the two big offenders, the two big mutagens. Okay, another example is a point deletion. So my point here is precisely that it's a single base or a single base pair that has mutated. May have drastic effects. In the case of deletion, okay, here is the original sequence of the DNA base, which through the mRNA codes for this particular amino acid sequence, okay, histine, all the way through. It's repetitive, CAT, you see it's a CAT. CAT triplet codes for histine amino acid, right? And it's all the same, but maybe this protein needs that many histines line up in a sequence. However, if there's a single mutation over here, let's say, in this particular A, and in this case, the mutation is going to be a deletion, not a substitution, but a deletion. In other words, this 1A is taken out of the frame, out of the reading frame. What happens? It shifts the whole, whatever is downstream, the whole code is shifted. And now, if there's 1A missing here, then this triplet is not longer going to be CAT, but it's going to be CT, and it's going to pick up the C of the next one, of this one here, because now this A is missing. So the new triplet you can see is going to be CTC, right? If this A is missing, the whole frame is shifted 1 over, and it changes the triplet code. One single deletion, 1 point deletion can change the triplet code. And it could be by changing that triplet code, in this example, all right, now we have the new triplet here is CTC, which codes for leucine. And then the next one is going to be ATC, and then it continues to repeat ATC, isoleucine, which is another amino acid, and it changes the amino acid sequence. All right? It's rare, but it can happen. And so point mutations can typically be either substitutions of a base or deletions of a base, and everything downstream then will change all the code. Okay? That's a single base or a single base pair mutation. Another possibility is that a segment, several bases, a few or a lot, all right? And there we have several examples. They can either be deletions, like in this example, this sequence here, C, D, E, F of this represents, this little bar represents a chromosome. That's the centromere there. And this is the chromosome. So the genes, let's say that these represent genes, A, B, C, etc. They're excised, they're taken out. And so this is a segment deletion, of course. That code is going to be missing in the mRNA, and therefore the protein is going to be defective. Or it can be a translocation, a movement from one chromosome to another. The sequence, a movement of the sequence. Or it can be a duplication. These are chromosome pairs now. This is a chromosome pair, right? A sequence, an arm, a leg of the chromosome is passed on to its pair. And so there's a duplication when this frame is read. It's going to have a duplicate, excess information. Or the other example is that the sequence can be inverted. All right, if the sequence is inverted, it can also affect the reading frame. And the end result, the protein can also be deformed or no longer code for that protein. So these are other examples of segment mutation, not individual points, not individual bases. Now, finally, the causes. Okay, so again, the three main causes of mutations. These causes are called mutagens. They're agents of mutation. They're mutagens. And they're also known as carcinogens because in the organism, in the individual organism, they cause cancer. Whether that is passed on or not is a different story. It has to be at the level of the gametes to be passed on, right? Okay, so the main ones, like I mentioned, is radiation, UV radiation, or chemicals, or heat. So three main types of UV radiation, A, B, and C. And this is why the ozone layer, which circles the Earth, is important because ozone, which is O3, anyone know the formula for ozone? O3.
O3.
Right? Okay, and it's gas, and it's unstable down here. It floats out into the upper atmosphere and makes that layer like a coating, like a shield of gas, of O3. It prevents UVC from penetrating. It filters out some of the UVB, but UVA goes right through the ozone. All right, so A and B are the ones to watch out for, and that's why sunscreen, you need to look at the rating. The higher the number of the sunscreen, the more it filters out the UV radiation. Here's an example at the molecular level, what UV can do. It can cause these dimers. This is one example. A dimer is to link two adjacent bases, not complementary base, not between the A and the T, because those are already paired up with these bonds. These are hydrogen bonds, okay? But rather adjacent. And so it will break the hydrogen bonds, which are relatively weak bonds, and it would link adjacent bases. Typically, it happens with thymine, and it's called a thymine dimer. When it does that, it kinks the DNA ladder, and it disrupts the structure, the actual physical structure. And that is, at the molecular level, an example of ultraviolet radiation, ultraviolet mutation. Okay, it tends to break the hydrogen bonds and link the thymines that are adjacent to each other. Okay, so that can cause mutations, obviously. At the chemical level, some chemicals are similar, look similar in structure to the bases and to the base pairs, actually. You notice the base pairs, if one base has a double ring, then the other base has a single ring. That's so that they all fit, because you can only have actually 3 rings total in between the rungs, right? In between the rungs of the ladder. The rungs of the ladder are parallel to each other, and they have to stay at the same distance from each other. So there can only be a total of 3 rings in the middle. Okay? These are the rings we're talking about. So this base is a double ring, and this base, the complementary base, is a single ring, right? Okay. And so if this is a double ring, then this one is a single ring, and so on and so forth. So it's a total of 3. Well, look at this molecule, which is called acridin. It's got 3 rings all linked together, so it fits right there in the middle. But again, it kinks the DNA, and it's disruptive, because when the mRNA is made, either it truncates here, or if this is red, it's going to misread the entire reading frame. Okay, so this is an example of a chemical that is a mutagen, that is a carcinogen, that can cause mutation in the DNA because it intercalates within the DNA structure itself. All right, and there are a number of these chemicals around. And finally, the heat issue is typically at the level of this other link. So there are 3 main chemicals here. On the outside, these balls, all right, are these circles. These are phosphate bonds. These are phosphate bonds. And then this blue ring, either the blue ring or the red ring, those are sugars. It's a type of carbohydrate. It's a pentose because it has 5 carbons, all right? So you have a phosphate and a sugar backbone. And then the bases, the actual bases are these. They're called nitrogenous base because they have nitrogen in this ring. They have the nitrogen element. Okay. And this bond, this little blue bond or this little red bond, okay, that is called a glycosidic bond. And it's an N-glycosidic bond because it connects to the nitrogen. I'm giving you excess of information here. But the point is that this bond, which is called an N-glycosidic bond, is subject to heat. All right, and can be disrupted by heat. And when it does that, what happens to the ladder? The ladder comes apart. Okay, it loses its integrity. So the end result of heat, of excess heat, is that the ladder, that DNA is denatured. The ladder comes apart. Now, of course, it's pretty excess heat. We know that the DNA can tolerate at least 98.6 degrees Fahrenheit, because that's the normal body temperature, and more than that. But if you heat DNA to an excess, the ladder comes apart and denatures. All right. Typically, by that type of heat, we move away from that heat source, but it can happen. So those, again, are the 3 main causes of mutation at the physical chemical level of the molecule of the DNA, and therefore the messenger RNA that will translate into the protein. That's about mutations, right? Okay. Moving forward now, let's look a little bit about recombination and then transposons. Recombination is also known as crossing over, and this is exactly what happens with the chromosomes. Here we have, what do we have here? This blue and this red. What is this blue thing? This is a chromosome, right? But you notice, and this is the other chromosome. They look alike, so these 2 are a chromosome pair. They're a chromosome pair, and they are homologous because they are the same. They have essentially the same information. So they have the same basic genes contained here. Let's say if here's the gene for hair, and here's the gene for hair, and here's the gene for eye, and hand, and foot, etc. not stomach, not heart, but those 4 genes, okay? So the chromosome pairs, one is represented in blue, the other one in red, and they're homologous because they have the same genes. They're paired up because it's a duplicate, it's a safety mechanism. If one pair dysfunctions, the other pair can pick up. All right, they are aligned, they're next to each other, so this cell is about to reproduce. First, the DNA is in chromosome shape. But also, you notice that this chromosome is X-shaped, right? So this chromosome has already duplicated. This one chromosome, the blue chromosome, has already duplicated its information because this was the original chromosome here, and it duplicated into here, and it's been held by this little structure called this centromere. It's just held together. But this is actually a duplicate chromosome. And because there's a total of 4 legs, it's called a tetrad. And then this pair, its pair, let's say that this is chromosome number 1. Its pair has also duplicated already through the process of what we know as replication. And so we have 2 tetrads. When the 2 tetrads are lined up at the equator of the cell, just before the cell splits, as you can see down here, all right? Some legs will cross over, okay? When they cross over, because this blue leg is a duplicate of this blue leg, and this red leg, the ones that are crossing over, is a duplicate of this red leg. Essentially, the genes that are here are essentially the same, right? But there can be some minor details in difference. There can be some differences at the level of minor details. That's what we call alleles. Don't worry too much about it. The general information is the same, but there's crossing over here, and the end result is that this blue chromosome ends up with a piece of the red chromosome, and the red chromosome ends up with a piece of the blue chromosome, right? The legs that crossed over are called recombinant legs, And the legs that did not cross over are non-recombinant because they did not recombinate, they did not mix. All right, if we look at, this is at the chromosomal level. Now let's look at the cellular level, or at least at the nuclear level, right? Actually, the cellular level. This circle represents a cell, and these are the precursor cells. In other words, in the female, what are these cells? Where are they? These are the immature oocytes in the baby girl, in the ovary of the baby girl, okay? These are the female oocytes. In the man, what are these precursor cells? These are precursor cells on the inner lining of the seminiferous tubules inside the testicles, which will produce the sperm, all right? Those are the precursors. Those are the key cells that need to watch out for mutation, because if either one of the 3 mutagens hit them, what are the 3 mutagens that can cause mutation? Either UV or chemicals, carcinogens, or heat hit those precursor cells, then it can cause a mutation, right? Okay, so these are the precursor cells. These precursor cells are getting ready to undergo meiosis, which is the process of making the gametes of gametogenesis, all right? So I mentioned it's like a double mitosis. Here's the first mitosis, if you will, and then there's a second mitosis. It's a double split. You see the first split here, you see a further split here, all right? So the first thing that happens is this one chromosome and its pair duplicates. So here are the duplicates, all right? Here's one chromosome and here's the other chromosome. So here's a chromosome pair, all right? How many total chromosomes in this precursor cell? How many total number of chromosomes in this precursor cell? How many total number of chromosomes? 2. How many chromosome pairs? 1 pair, all right? This is one, let's say it's chromosome 1. This is 1 pair of chromosomes. 2 chromosomes, 1 chromosome pairs. How many individuals make a pair? 2 individuals make a pair, right? Okay, all right. See how each chromosome has duplicated, but they're still stuck together by the centromere, all right? So this is the tetrad. Now they have lined up at the equator. You see aligned? They have lined up at the equator of the cell, the center of the cell. Now crossing over has occurred here once the cell splits in the middle, and each pair has migrated to the pole. This is the North Pole, this is the South Pole, all right? Before the split, what has occurred between here and here? In diagram B, what has occurred between here and here? What has occurred? Crossing over has occurred, because you see that this blue chromosome has a piece of the, I'm sorry, this red chromosome has a piece of the blue, and this blue has a piece of the red. So between here and here, what happened? Crossing over, this happened, all right? And then after this happened, crossing over cannot happen after the split, right? Because then the pair is no longer around. Okay, so the crossing over happens at the alignment at the equator. Then the chromosomes are pulled either to the North Pole or the South Pole, and then the cell pinches in the middle, and you end up with two cells. We're halfway. This is meiosis. This is meiosis. It's not just mitosis, it's a double mitosis. So we're halfway. This is the first level. But you see, at this level, the crossing over has already occurred. So recombination has already occurred. So there's variety has been introduced, okay? Variety has been introduced by the recombination event. Then the second split is a normal split. The second one is like a regular mitosis. In other words, this one cell will split into two. What happens? What needs to happen to the centromere? The centromere needs to break. Centromere needs to break to release the duplicates, right? To release each arm of the duplicate, so that now one set will go to the North Pole, the other set will go to the South Pole, and the same happens to the other cell, all right, with the other recombinant. And the end result of gametogenesis is that you end up with 4 cells from how many original? One original cell. You end up with 4 cells because it's a double split, all right? And of those 4 cells, 2 of the chromosomes are identical to the original. 2 of the chromosomes are identical. You see fully red and fully blue. But the other 2 are what? They are recombinant chromosomes. They are recombinant chromosomes because this red has a piece of the blue and this blue has a piece of the red. So the variation is in these 2. Let's say that these 4 are eggs. These are 4 mature eggs. But the woman only ovulates how many eggs at a time? Normally, only 1 at a time. From these 4, only 1 is going to mature, become the 1 mature egg. It could be either this one or this one or this one or this one, okay? If it's this one or this one, at least on the female side, the female information of that little embryo, the 50% female information of that little embryo is going to be identical to the mom because of the precursor cell, okay? But if either this egg or this egg gets fertilized, then it's going to be the recombinant information. It's going to be the recombinant information.
So what will be the effect here or the difference?
What is the difference? The difference can be subtle but real. It could be a difference in height, for example, you know, or it could be a difference in the amount of melanin, or it could be a difference in the shade of the eye, right? It's subtle but real. So the difference between the red and the blue, because they are essentially identical, all right, but that essentially means that there's slight variation. This is where variation occurs, at the chromosomal level. It's at that level of detail. It's amazing, it's fascinating, and we call it elegant because this happens without our knowing it, without our willing it, in every species of the planet that reproduces sexually.
So
amazing.
Off track on this one. If someone chose, so this affects cloning.
Well, but remember now we're talking recombination is in sexual reproduction because we're producing gametes, right? Which is only for sexual reproduction. Cloning is simpler because it's just a simple it's a simple mitosis, a simple split. So whatever happens to the nucleus of the parent cell, that will be passed on average to the two daughter cells. Okay? So cloning is actually simpler than this because this involves production of gametes first and then fusion of gametes second. All right? And the odds are against all of that happening. And that's why there's excess. All right, so basically, I've probably given you a little too much detail, but I want you to understand recombination or crossing over, that it's a natural process. It's happening in our cells at the duplication level, at the level when the chromosomes duplicate and then cross over when during gametogenesis. In other words, for men, during the production of sperm, and for women, during the production of mature eggs. Right? Okay. There's another type of variation that can also be introduced by what is known as independent assortment. Independent assortment. Let's start here with the parent cell. All right. And this, again, is how do we know that this is meiosis and not just mitosis? You notice, and the parent cell, this is the parent cell, right? This is the parent cell because the arrows come out from this cell. So this is the parent cell. How many chromosomes in the parent cell? 4 chromosomes, exactly. How many pairs? 2 pairs, right? There's a large pair and a small pair, right? But my point is that are the chromosomes in the parent cell paired or unpaired? They're paired, right? Red has its blue and red has its blue, so they're paired. How about these cells down here? This is a cell, this is another cell. Are these chromosomes paired? They're unpaired. So that gives you a hint that these are what? These are gametes because they're unpaired chromosomes, all right? And therefore, this process is not just mitosis, this process is meiosis because meiosis is what produces gametes. So they're unpaired, that's the first hint. And also, how many daughter cells do we end up in either one of these processes? Either possibility A or possibility B. How many daughter cells are here?
4.
4. And the original parent cell is 1, right? So when you have 4 coming out of 1, then it's meiosis because there's been a double split, right? So all this to say that this is occurring in meiosis. All right, so remember that these chromosomes first need to line up at the equator, all right? When they line up at the equator, you have 2 possibilities. This is one possibility, how they line up at the equator, all right? In fact, the cell is, the problem with diagrams in science is that they're not too well done sometimes. The equator is actually represented here vertically, all right? The equator is here, not horizontal, but vertical. So please think of the equator vertical like this, all right? Rotate the Earth 90 degrees. The equator is vertical here, all right? That's the equator, and that's the equator here. And therefore, on this cell, this is one pole, and that's the other pole. This is one pole, and that's the other pole, right? Okay, so these are 2 possibilities of lining up at the equator. Either they line up like this, so all the blues are on one side of the equator and all the reds are on the other side of the equator, or they can be mismatched. You got one blue and one red, and you got one red and one blue. Those are the 2 possibilities, right? When these cells migrate to the poles of the first set of daughter cells, if they're lined up this way in possibility A, then the daughter cells end up like this. You got all the blue chromosomes in one cell and all the red chromosomes in the other cell. But if the lining up at the equator is like this, then one daughter cell will end up with a blue and a red, and the other daughter cell will end up with a red and a blue. So there is this other possibility, the gametes will come out differently. The gametes will come out differently. Remember, these are two sets of chromosomes, okay? So the gametes will contain two chromosomes. In possibility A, the end result is that half of the gametes will have blue chromosomes and the other half will have red chromosomes. They're pairs, so they are essentially identical, but there's slight variation difference, all right? They carry the same genes, but within the gene, there's variation. Remember, there's variation always, the quantitative traits. In the second possibility, the chromosomes are going to be interchanged so that all of the gametes will have one blue and one red, one blue and one red, one blue and one red, one blue and one red. So you see, there's greater variety in this possibility. There's greater variety here, okay? And this is what is meant by independent assortment. The assortment is here. They have been sorted out from the parent cell to the first daughter cells. The chromosomes have been sorted out. Okay, it's like a deck of cards. You have half the cards going this way and the other half going that way. They have been sorted out independently, okay? In possibility A, all the blue chromosomes are staying together and all the red, therefore, are also staying together. But in this other possibility, it all depends how they lined up at the equator. You can end up with variety, okay? The lining up at the equator, this lining up at the equator is independent. There's no reason why all the blues have to be on one side of the equator and all the reds have to be on the other side of the equator, because these are essentially identical chromosomes. So they could be on either side of the equator. It's a chance, it's a 50-50 chance. It's just how those chromosomes got to the equator, all right? If they're on the top or the bottom part of the equator, on one side or the other side of the equator, all right? And so that's a 50-50 chance that we have possibility A or possibility B. But the end result is drastic difference. In one case, there will be less variety than in the other case, all right? And that's why the assortment, like I say, occurs at the first division here when the chromosomes are sorted out. And because they lined up independently, then this process is called independent assortment. Independent assortment. And then the last thing I need to tell you is that each one of these pairs is called an allele. All right? Each one of these pairs is called an allele. I showed it in greater detail. I thought I had a slide. Oh, yeah. Further down here. I'll show you alleles again later here. All right? But let me anticipate for a moment. So we talked about how many chromosomes here? Two chromosomes. How many pairs? One chromosome, because they look alike. All right? But have they duplicated already?
Yes.
Yes, they duplicated. All right? So here's a gene. Gene A codes for the color of the shell of this ladybug. All right? And capital A codes for red. And lowercase a codes for yellow, or whatever the cream color that is.
Yeah.
All right, so that's what the gene is. So you see these are alleles. This is the dominant color is A, and the recessive color is small a. It's the same gene, essentially the same gene, but there's a slight difference in the color. Okay, there's a this is variation in the color. So these are called alleles. This is the dominant, these are the dominant alleles, and these are the recessive alleles. Just technical terms. The following part is a little more fun, less dry than this one. This one is very technical, but these are alleles. So here's one allele, this is the other allele of the chromosome pair. And that's why independent assortment of the alleles in this case. That's another source of recombination. That's another source of mutation or variation, which can may lead to evolution, to speciation. All right, let's look briefly at transposons. Transposons are segments of DNA that jump around. It's amazing the dynamics that go on in the DNA. This is within the nucleus, within the DNA, within the chromatin structure. Sometimes these segments jump from one place to another, or they can invert, or they can jump. There's a little video, and I don't know if this is actually active. I will try it. Yes. Okay, hopefully not too loud. Right there.
Transposable elements, which are found in virtually all cells, are segments of DNA that can move from one location in the genome to a target sequence in another. A simple type of transposable element called an insertion sequence, or IS element, is only 700 to 1500 base pairs long and consists of two basic parts: a coding region or enzyme called transposase, and two short inverted repeat flanking sequences. The cellular machinery transcribes the transposase coding region and translates it into the transposase.
So this is DNA, double helix,
Transposase
messenger RNA,
transposition process.
making a protein.
The transposase randomly selects one of many possible target sequences.
The protein selects a sequence.
The IS element, depending on the particular type, may enter the target sequence by one of two general mechanisms: replicated transposition or non-replicated transposition. In replicated transposition, transposase nicks the transposable element, and the nicked ends attack the target
They nick,
sequence,
they cut.
making a staggered cut in the target DNA.
Staggered.
DNA polymerase fills in the large gaps, and DNA ligase seals the remaining nicks. A recombination event resolves the two molecules. As a result of replicated transposition, the cell has a copy of the transposable element flanked by duplicated target sequences.
Okay, so all this occurs with a very complex machinery at the molecular level of proteins, starting with the original DNA sequence, which is a double helix here, as you can see, flattened out for simplicity. But this is the one that codes, this red region is the one that codes for that protein, which goes into another sequence of the DNA in a different region. It could be of the same chromosome or a different chromosome, and essentially make a cut and transpose, pass the one DNA sequence into the other region of the chromosome, of the chromatin, and duplicate that sequence in another region of the chromatin. Sorry, question.
So this is just one way of transport.
Yes, it's one of the two mechanisms that is called replicative, because replication is involved. In other words, when you see replication, think of duplication. The DNA duplicates by replicating. Now, there's another mechanism that has been discovered.
Transposition is sometimes called cut and paste replication.
Cut and paste.
In this process, the transposase brings inverted repeats and flanking
It hairpins it, hairpins the DNA.
One phosphodiester bond is cleaved on each strand at opposite ends of the IS element.
These are proteins holding
OH groups.
the complex.
Each 3' OH
It's a nick here.
attacks the other strand of the double helix, creating a new bond that results in hairpin structures. Post-carrier DNA is ejected, and the hairpins on the IS element are re-nicked and attack target DNA.
Inserted here. All this happens by a machinery of proteins.
From the 3' hydroxyl groups, the cell's DNA polymerase molecules fill in the gaps.
The last step is to seal the gap.
Jumps from one DNA molecule to another. To summarize, in non-replicated transposition, the transposable element is cut and pasted from one DNA molecule to another. In replicated transposition, the transposable element is copied to yield two transposable elements, one in the original position and one in the target DNA.
Okay, so this is luxury of details. Really, I don't expect you to know this level of detail, but at least to know that this transposition is segments of DNA from one region of the chromatin inserted into other regions of the chromatin, right? That will definitely affect the target region. It will affect the reading frame, the coding on the target region. If this original purple protein was a single read, that's going to be affected. Now it's going to have this other read inserted in it. And these transposons happen in different parts of the DNA. It's estimated that about 40% of the human genome is made up of transposable elements. It's a very significant amount, all right? Okay, that's it. Stop. Please stop. Thank you. All right. There are other, if you go there online later, there are other movies about transposition, but you get the basic idea is that DNA jumps around through very complex mechanism inside the DNA. When that DNA is inserted, it disrupts the original sequence that was there. This may have originated through a process through viral infections. How did we accumulate 40% of our DNA that is transposable elements? In other words, foreign elements that have been inserted into our genome over the millions of years that the species has been around. And this happens with essentially any organism. It is thought that it has been through viral infections, okay, viral infections. So let's look at a viral infection for a moment. Viral infection has a cycle, has two cycles. One is called the lytic cycle, the other one is called the lysogenic cycle. So just to be clear, a virus is essentially a structure, a chemical structure that has a protein code, and inside the protein code has either RNA or DNA, all right? But they look like satellites. They look like these machines that are very structured, okay? And they're either spheres or capsule-like structures. They are much smaller than bacteria, which are much smaller than cells, all right? Millions of bacteria fit in every one of our cells, and millions of viruses fit inside a single bacterium. So these are super small. Here's a scale, for example, 100 nanometers. A nanometer is 10 to the?
-9.
-9, -9, right? 10 to the -9, 1 billionth of a meter. All right. So these are some structures. These are actually electron photomicrographs. Here is the process of infection. A virus is also called a phage in the US or a phage in British. But the DNA phage, this is a phage, this is a virus that contains DNA and not RNA, which means that it's going to be double-stranded. When the infection occurs, that DNA, the viral DNA, is introduced, inserted, in this case, into a bacterium, or it could be a human cell, all right? But in this case, it's a bacterium. So these are called bacteriophages or bacteriophage. Here's the bacteria, here's the chromosome of the bacteria. Now, this DNA of the virus can form a little circular DNA, which we call a plasmid, right? Little circular DNA. Is still distinguished from the bacterial DNA. But now this bacterium has two options. Either it goes into what is known as the lytic cycle, or it goes into the lysogenic cycle, right? And all that is geared by proteins and by chemical reactions and so forth that is monitoring the process. But if it goes into the lytic cycle, what happens is that this plasmid, using the bacterial machinery, this plasmid will replicate itself and will produce both more DNA, viral DNA, and also the viral coat, the protein coat, whether it's a capsule shape or a spherical shape. All of the structures of the virus will be reproduced by sequestering the machinery of the bacterium cell. That's why they are necessarily parasitic. Okay?
It's one or the other, Father.
It's one or the other. One or the other. Yes, there are chemical issues and environmental issues that determine which way this cell is going to go. All right. But let's stick with this lytic cycle for a moment. If it goes through the lytic cycle, what we have is that the DNA of the virus will make more of itself and more viral capsules, and you end up with a bacterium cell that fills up with viruses. At some point, it's so full with viruses that the bacterial cell explodes, all right, and releases the viruses, propagating the viral infection. That breaking up of the cell is called lysis. When the cell breaks up, it's called lysis, all right, and it releases the virus, which will then proceed to infect other bacteria in the vicinity, or in the case of an animal or a plant, other cells within that organism. So this is the infectious cycle here, all right, the lytic cycle. Again, the DNA of the virus uses the machinery of the host cell to generate more viruses, which will then break out of the cell and infect. Each one virus can infect one more cell. So it's a rapid progression of the viral infection, right? That's one alternative. The other alternative is to go into what is known as a lysogenic cycle. Essentially, the viral DNA goes hidden, hides, inserts itself into the bacterial DNA, or the human DNA, or the host, whatever host this is. This can be a plant, a fungus, an animal, or bacterium, or archaea. All organisms are subject to viruses, okay, to greater or lesser extent. All right, so it goes into hiding. In the lysogenic cycle, the viral DNA inserts itself somewhere on the bacterial DNA. At that stage, it's called a prophage, just a technical term. It's called prophage. Then that bacterium doesn't realize it's got this DNA stuck to it, viral DNA. So when the bacterium divides by binary fission, all right, that DNA gets replicated. The bacterial DNA and the viral DNA gets replicated and passed on to the daughter cells. And many cell divisions, all of the bacteria that come from this infected bacteria will have, in principle, will have that viral DNA sequence there, which at some point, typically there's an environmental or a chemical stimulus will cause that viral DNA to come out of the chromosome, to come out of sequence, and go into the lytic phase, into the lytic cycle, all right? So you end up with this horizontal A shape, figure 8, all right, where the virus cycles either into a lytic cycle or into a lysogenic cycle, back and forth like that, back and forth, excuse me, so that at the same time, you know, within a relatively short period of time, the virus can infect many other cells of the organism, or the virus can go hidden and come out the next day, the next month, the next year, the next decade. Like, for example, in HIV. Goes into this phase, hidden, and can come out a decade later and cause the infection. All right? Lysogenic. So you see, this is actually the beginning of a transposon.
You mean undetectable?
Right. Now, there has to be some signaling at some point. What's the only thing that's going to kill this is the antibodies of that the body itself produces. The body detects that this cell is sick, and the antibodies attack the cell and essentially kill that cell. So it's a self-killing process, all right? Because those are infected cells. Mm-hmm. So thank God that most of these viruses are neutral. They don't affect us much. And what we have, the current theory is that all these transposons that we have in our body, this 44% of our genome, that's almost half of our genome, okay, of the 3 billion base pairs, almost half of those 3 billion base pairs, are possibly ancient viruses or fragments of viruses that have been left over in our bodies over millions of years, that are there and get replicated and get passed on from one generation to the next. Thank God they're neutral. Sometimes Many times they can cause infection, and sometimes are actually positive. You know, they create because they generate variation, and at some point they may actually code for antibodies or things like that that can help prevent other infections. So it's a very, very dynamic, complex system that is all happening at the molecular level, all right? And the whole point here is that transposons may also cause mutation, may cause evolution over a long period of time. But the whole point here is that this is at the DNA level, all right? It's at the DNA level. Okay. A big summary in one slide, that the first cause of mutation that we're calling mutations in general can actually be mutations on the DNA, or it can be by recombination event during gametogenesis, or it can be by transposons, movable elements of DNA from one piece of the DNA to another, one segment of the DNA to another. Okay, that's a big summary. We've been very patient. I'm going to stop here for a few minutes, and then we'll pick up on the other two sources of variation of evolution, which are migration and drift at the level of the organism, at the level of the population. Okay, so I'm going to pause the recording for a few minutes. If you can take whatever you need, 3 to 5 minutes, something like that, and then we'll continue again. All right? Any questions or comments up to now? People are good? All right. Okay, welcome back, and hopefully it's recording again.
Yep.
Great, thank you. So now moving into the second and the third force or cause of evolution, which is migration, the second, and the third one is drift, genetic drift. Now, of course, they don't have to occur in sequence, all right? They may occur simultaneously, or any one or any combination of these forces or factors: migration, mutation, drift, and selection. But actually, I've mentioned several times, the one that really actually physically causes evolution or may cause evolution is selection itself, which is essentially selecting out the weaker, the less fit. That will be covered next lecture in fuller detail. For now, I want to concentrate on this other force of evolution that is known as migration or gene flow, all right? Now, I mentioned that this is at the population level, and it's also at the individual level. It's just that in nature, no individual really lives alone. You don't really find any individual out there living alone. They are typically interacting with other individuals of the same species. And the minimal interaction is going to be between how many? Two. So two individuals of the same species make a population. A very tiny, small population, but it's a population. And I'll show you a graphic example of that in a few minutes. And it can be as large as millions, for example, millions of bacteria in this one colony. And I'll show you another example of that also in a few minutes, okay? But be clear that whatever individuals is in that population is of the same species, and they are interacting. Then when populations interact among themselves, or between themselves, populations of different species, then that forms what we call a community. Perfect. All right. So this is the gene flow. This is the gene flow within a population. And the flow is called migration, and there are two types of migration. Either the genes are flowing into the population or the genes are flowing out of the population, right? Now, who's carrying those genes? Well, the individual is carrying the genes. You and I are carrying human genes, and we move in and out of several populations, our family population, our work population, our study population, right? And so wherever we go, we're carrying our genes in and out of those populations. So there's either immigration or emigration of the genes. But of course, it's the whole organism, the whole living organism that is carrying that package of genes. So that's why it's called gene flow. So here's an example of emigration All right, need a little imagination, but basically you can see that all these zebras are moving in the same direction, right? From right to left. So we can assume that they're leaving a patch of grass over here and going to another patch of grass over there. So they're emigrating. Whatever genes, this population, this is a population of zebra, maybe several dozen zebras, all right? It's a population, it's a herd in Africa somewhere, and they're moving from one geographical region to an adjacent geographical region, one patch of grass over here, which they grazed already. Now they're going to go into another patch and graze that one. So there's an emigration of whatever genes this population of zebra carries from this patch of grass into the other patch of grass. All right, so as you see, it's at the level of the organism, it's at the level of the population. Okay, now, just to highlight variation a little bit, this insert down here is a close-up of the 4 zebras in the middle. 1, 2, 3, 4. You see these 4 zebras in the middle? You could say they're essentially identical, right? The pattern is certainly a black and white pattern, and it's mostly vertical stripes, black and white. So we can say that they are essentially identical, they're generally identical, but where is variation? Look at variation. All right, for example, see, here's a black and white pattern, right? Black and white, and this other one also black and white. This one black and white, but it's got some in the middle, in between, lighter black. All right, lighter black, extra. It's not in this one. It's not in this one. And look at this one. Very pronounced in the middle black. In the middle black, it's like double striping, right? It's a double striping pattern. So this pattern is really not 100% identical to this one, or this one, or this one. So there's slight variation in the pattern. Also, look at the dorsal area here in the back area. Look at this pattern here. Concentrate where it changes from straight vertical to diagonal. Look at the pattern here. Slightly different from similar, but slightly different, not exactly identical. For example, there's this V shape here, similar to this one, but not identical. And there's another V shape here that is not really on the same place here. See? Slight variation there. Now look at over here. Where's that V shape? Not really there. All right. And this one, again, different. You got 4 different patterns on the back where the pattern changes from vertical to diagonal. These 4 different, that's only 4 of them, even though in the general they all look very similar alike. That's called mimeticism. That's to throw off what's the advantage of this pattern. Now this is a photograph, right? So it's a still motion, but these animals are moving. They're moving sideways, so this vertical pattern is moving sideways. And these are all individuals. When the lion is trying to hunt down, the lion is not going to hunt down the whole population, right? The lion needs what? One individual. How's he going to focus? How's the lion going to focus when all these patterns are moving around, you know? And he needs to focus on one of these and take that one down. All right? So it's a pattern, it's a camouflage pattern that helps the individual blend into the whole population, right? So there are evolutionary advantages there. If any one of these zebra comes out with a drastically different pattern, let's say that all its stripes, it's also a black and white pattern, but let's say that they're all horizontal. It's going to stick out, that one's going to stick out, right? The lion says, okay, that one's, I can focus on that one. It's going to go for that one. That's not going to survive. So the selection pressure is going to be on having a very similar pattern, but with tiny variations. Because then the zebras themselves would distinguish between these 4 guys. Okay, and maybe one of these is a male and the other 3 are females. All right, so the zebras can distinguish between their variation, but the lion is all confused. So you see? Anyway, the point here is that the gene flow is of the patterns that these zebras carry, in this case, out from this geographical region into the other geographical region where they're going. Doesn't only happen in animals, it happens also in plants. Here's an example, and I can tell you that there are two populations in this dune. This is a sand dune. Here's the Atlantic Ocean. Here's Key Biscayne, or whatever you want to call it, all right, over there. And this is sand dune, right? Okay, what are the two populations of plants here that are obvious to the eye? See a little tuft of grass here? And see another tuft of grass here? Another tuft of grass, tuft of grass. They all look similar. We can assume that this is the same species, right? But this is one individual tuft. Is this another individual tuft? And another one, another one, another one. Okay, so there are several tufts of the same species. So this is a population of grass over here. This patch is a population of grass. We don't know how big it is. It could be small, it could be large, but at some point there's going to be empty sand in between. So this is a population of grass. And by the way, if you look closely, you can actually see the little shoots. How is this grass reproducing here? See, little runners. See here, the little runners, horizontal. What is that called?
Vegetative propagation.
Yeah, vegetative propagation, right? Little shoots, little runners, and new tufts coming out. Okay, so anyway, there is a population of grass here that is growing in what is otherwise barren sand. Okay, this, by the way, is called a pioneer species because it's coming into an area that has no other species living on it yet, at least at the microscopic level. But then there is this other plant back here, right? Now, is this a grass? Look at the edge. It's got runners, right? So this looks more like a creeper. This is not a grass, it's a creeper or a vine, okay? But, and it's safe to assume that this whole patch here of green is not just one individual big plant, all right? There are probably many individuals all intertwined in here. So here's another population of a plant, in this case, a creeper, not a grass. So that's why I'm saying this is a community, because this is one population. This is obviously a different species than this, right? All right, two plants, they're plants, but they're two different plants, so two different species. This is one population of grass. This is a population of creeper, which looks like this is the same. I think it's safe to assume that this is the same plant that this one, the same species.
Although it looks a little bit darker.
Well, that could probably the way is further back and maybe the way the sun is hitting it and so forth. But it's fairly safe because this is a pretty barren environment, right? So you would expect relatively few species to be able to live here. What's your intuition? Is this, does this have an excess of water underneath, or is the water going to be difficult to here? To obtain water, it's going to be very dry, right? So you could expect underground, under the sand, what do you expect the root system of these things to be? Extensive fibrous root system deep down to tap into whatever little water molecules they can find, all right? And it's true. Okay, what I'm trying to say is that most likely this species is the same as this species, but you see that they're divided here. There's a path all the way out to the ocean somehow. So this is one population of the creeper. This is another population of the same creeper. Thank you. Okay, this is another population of the same creeper. So we have a small community here, which has one population of grass and two populations of the creeper, the same creeper. Anyway, what I wanted to point out is that these are immigrating. These plants generally are immigrating. In other words, you see how the tendrils are coming out this way, the little runners? So this population is immigrating, is invading what is otherwise barren territory. You have to think very locally here. This edge of sand is otherwise unpopulated. So this patch of grass or creepers is immigrating into this region. And the same, these grasses are extending generally toward this area that is barren. See how the shoots are coming this way, little runners? horizontally, see? All right, so what I'm trying to point out is that here's an example of a plant, not an animal, that is immigrating into a new region, all right? Local, but it's a new region. So these plants are flowing, their genes are flowing into a new region. Immigration, emigration together do migration or gene flow. Forward. So we can find that there is a vast range in the number of individuals in the population. There's a vast range in the territories that they occupy, the geographical territories, and so forth. So there's a wide variety of ranges. There's vast range that happens, both in plants, in animals, in fungi, in bacteria, and in archaea, all right, in all the domains. Here's an example of the smallest possible population. What's the smallest possible population? 2. Anybody know what these are? They're called sloth. Yep. They're from the anteater family. They're from South America. The anteater lives in Africa. And so that's when the continents divided. They diverged. So it's an example of divergence. But they have common ancestors. And the key relationship between a sloth and an anteater is what? The big claws. The big claws, all right?
They also know to be very lazy fathers.
Yes.
That's why they don't immigrate too far.
Exactly. You got it. You got it. That's exactly right. So here's the anteater. And this is the claw of the anteater. Right? Big claw. In this case, the claw of the anteater is used for, they also have a long tongue that's sticky, for digging into ant holes and termite holes. Whereas the claw of the sloth is used for hanging on, hanging on to branches. All right, and so the sloths are classified into three-clawed sloth. It's like the tongue twister. The three-claw sloth. Okay, see that? The three-claw sloth. Three-claw sloth. Three-claw sloth. Three-claw sloth. Or the two-claw sloth. Okay, and this sloth is an example of a three-claw sloth. All right? If you have two, it would be an example of a two-claw sloth. That's how they classify it. But this is a very tiny population of a mom and her baby hanging on for dear life with a nice smile for the camera in South America somewhere. So they move very slowly, and it's done for conserving energy, except when they're attacked, and then they can run very fast for a short period of time. But they tend to hang up high. And when I lived in Panama for a little while, we'd be on the road in the countryside, through the jungle, and sometimes they cross the road. You have to be careful because they take about an hour to cross the road, okay? They go real slow. And then you go further up the road, and then you can actually buy them because the young children in the area who are typically poor, they catch one of these, and then they strap it onto their body, and they just stand there on the road until someone comes by and buys it from them. Yeah. So it's a whole local economy. But they're gentle, they're mostly vegetarian, eat leaves, and so on and so forth. But here's an example of a very small population of a mammal that doesn't move more than maybe 1 mile or a few trees from the place that we were born for their whole lifetime. For the entire lifetime, they will just hang around the same trees. Contrast to that, of course, populations are not only animals, populations are also plants, right? And so here's a population of a pine forest that is very vast. It has thousands of individuals represented in this one population that extends from valley to hills and so forth, and has a very wide range of dispersal. Not the plant itself, not the adult plant, because what happens with the adult plant? The adult plant is sessile. In other words, it's stuck on the ground through a root system. So the adult plant is not going to move around the mountains. What moves around the mountains? The seed of the adult plant. The seed, which are winged, all right? So it's a wind dispersal. And the wind dispersal will then populate an entire mountain range of essentially the same species of pine, a climax community of pine forest, because the seed of the pine, in other words, the embryo of the plant, gets dispersed by wind over a wide range, okay? So these are examples of the kind of counterintuitive. Here's an animal that moves very little, and here's a plant that moves a lot through its embryonic stage. So you have to think in those terms. Huh? All right, moving forward. There's actually a dynamic state of flow. That's why it's gene flow. Constantly, the populations are in a dynamic movement. No organism is just standing there, sitting still for any period of time. There's a flow in and out. By the way, there's a flow, there's an immigration and emigration of a third individual in this population. I told you that the sloths are mammals, right? They're obviously mammal. They have hair. And if they're mammals, how do they reproduce? Sexually. And you would expect this baby to be clinging on to its mom or its dad? Mom, because it lactates, because it's a mammal, and it's a mother that has a functional breast. And so there's a third individual of this population that migrates in and out periodically. Who is that? The dad, the male. Okay, so it's a third individual in this population that migrates in and out, and may also be a member of another population, a few branches away or a few trees away. So you see the flow? That's a very drastic example of the gene flow. The female genes are more restricted, but the male genes are going to get around to different populations, potentially, right? And that's a standard in mammals and many other species. All right, let's look at, so there's a dynamic state of flow. Here's an example of plants on one hand and bacteria on the other. So here's a patch of daisies, looks like, wildflowers, that is covering this hill. Potentially thousands of individual daisy plants form this population, right? Okay. The core of the population tends to be stable. In other words, the genes in the middle of the patch tend to flow less than the genes on the edge of the patch. All right? And so the periphery is more subject to variation. Yeah.
Probably because the center of it, the core, has less room to manipulate it.
Exactly. It's more established.
Yes.
It's more established, all right? Whereas the edge is going to be involved in emigration, in moving and expanding the territory. But as it does that, it's going to encounter more difficulties. Maybe here there's less water, maybe there's more water than in the middle of the patch, okay? Or maybe if there is a herd of deer coming this way, they're obviously going to start grazing where? On the edge instead of the middle, right? They found, they love daisies, so they're going to start eating the first daisies they see. They don't care. They're not going to go start in the middle just to disrupt the core, all right? So the edge in general, the periphery, is the one that is more subject to variation. Now, this also happens across the board, in any particular population that you can think of. So here's another drastic example of a microscopic population, bacteria. This you recognize as a Petri dish, and each one of these spots or blobs, these are colonies. These are individual colonies that come from a single individual bacterium that started reproducing by sexually or asexually? Asexually, by binary fission, right? By growing and splitting and growing and splitting, maturing and splitting, maturing and splitting, doubling up. So it's a doubling of the population. This is, for example, bacteria. And at every new generation, how many new individuals do you get? You get a new set of individuals, so it's a doubling effect. So I start with one bacterium. One microscopic bacterium was placed, let's say, on this patch here, on this column. That one microscopic bacterium eventually matured and split into how many bacteria? Into two. Each one of those two grew and split into two more. So from one we have two, from two we have four, from four we have 8, 16, 32, 64, 132. What you notice is known as a geometric progression, not arithmetic, but geometric. It doubles the population at each turn, okay? The next thing I need to tell you is that under ideal conditions, this happens for bacteria, for example, every 20 minutes. So you get 3 new generations every hour. Okay, in 24 hours, 3 times 24. So what started with one bacterium in 24 hours becomes a colony of millions of bacteria. They're all cloned. They're all identical, essentially identical in the DNA, unless some mutation has occurred in any one of these individuals along the way, which would have been passed on to that particular progeny of that one individual. Okay, so it's a geometric progression. It's extremely fast under ideal conditions. It's every 20 minutes, and that's why bacterial infections can advance pretty fast. There's a little anecdote of the fellow who invented chess, the game of chess, right? Which, you know, the square is 8 by 8. A square is by 8, all right? And the anecdote goes that he invented it for a Persian king way back thousands of years ago. And the Persian king, who was otherwise bored, was fascinated by the game and said to the fellow who invented it, who I think was maybe a mathematician, said, "Oh, Your Highness, all I want is a grain of wheat from your granary, right? A grain of wheat for every square." And so it's 8 times 8 is tables 64, right? Total 64 squares. So just a grain of wheat, but if you would please double it at every square, just double it. So you start with the first square 1, and then double again. And the king said, "My goodness, you're asking too little." So he asked the steward. The king said to the steward, "Go to the granary and give this man his wheat." All right. And when they started doubling and doubling, by the time they got to 64 doubles, okay, it was I don't know how many millions of grains of wheat, and all of the granaries in the kingdom were not enough to fulfill the payment. And so then it goes that instead of giving this inventor the grain, the king decapitated the inventor because he realized that the inventor was taking advantage of him, was fooling him. You know, but that's a geometric progression, can be very, very aggressive, very fast. All right, that's a bacterial reproduction. But then, so all this has happened. You see, in 24 hours, this is what we have from putting one individual bacterium in each one, where each one of these dots is. Then I go in 24 hours and I put some antibiotics here. So I put an antibiotic on the Petri dish, right, in liquid form. Which bacteria are going to be affected by the antibiotic first? The ones on the periphery. The ones on the periphery. So on the periphery, they're more subject to variation. And maybe the antibiotic starts killing the bacteria on the periphery. Now, they're all clones. So the antibiotic continues to kill the spot, but at some point it runs out of strength and maybe it doesn't get to the core. And so the core remains stable. All right, so that's an example. You know, you have to think of the factors that can influence this population. and how variation could be reduced, but the core will remain stable. Another example in migration is what we call a succession to a climax community, which starts, it's 3 steps, in the big picture is 3 steps. You start with the first step is the pioneer species. It's also known as the founder effect, all right? The pioneer species is where there was otherwise just barren soil or rocks or and decaying tree trunk, whatever, but there was no life on that region, right? That geographical region. It could be a small geographical region like the plate, the Petri dish, or it could be a huge geographical region like a whole side of a mountain, but there's no life there originally. Whatever species settles in there, that's the pioneer species. At the macroscopic level, typically it's plants first, and it's actually lichens, which is a combination, it's a symbiosis between algae and bacteria, photosynthetic bacteria, and fungi. Fungi and algae combine to form mosses, lichens. Algae and fungi combine to form lichens, all right? And so the fungus is a saprophyte. In other words, it feeds on decaying material, but the algae are photosynthetic, so they can provide some nourishment for the fungus, and the fungus provides a liquid internal environment for the algae in which to live. So you can see patches of lichens growing either on tree trunks that are decaying or even barren rocks. That's the first stage. Eventually, that generates organic material, decaying organic material, and then other species of plants can become established in the area. There's a case of immigration into a new geographical region that was otherwise barren. So you get little shrubs, little grasses, and so forth, herbs, which at first are annual. In other words, they flourish in the spring, they die in the winter, and come back again. And so in that cycling, they're dying and growing new individuals from seeds. So it's accumulating more and more organic material. Eventually, more plants can move into the area, immigration, gene flow in, new species coming in, forming communities of populations with stable plants, perennial, that will not cycle year to year, but live longer. Then you get shrubs, then you get trees establishing in. Essentially, eventually, you will get dominant trees coming in and forming what is known as a climax community of a dominant species. And I can give you one example for us in South Florida. You just look at the window there, and what you see there is all pine trees of one species, is known as Pinus elioti, otherwise known as the Dade County /pine. Dade County /pine is the dominant species of the climax community of South Florida forest, highland pine forest, all right? And that's at the tree level. At the level of the shrub, it's palmetto palm, the palm. So it's very interesting to see pines and palms growing together in the same geographical region, because essentially the pines are conifers. They belong to the north. They tolerate, they have needles and so forth. They're adapted to a northern environment, including snow, whereas palms are monocots. They're very intolerant of cold. All right, they live in tropic and subtropical region. So who has adapted here to the region, the palm or the pine? It's warm subtropical region, right? So it's native, it's okay for the palm. The one that has adapted is the pine. The pine, these Dade County /pine have migrated over probably after the last ice age. We're talking about 10,000 years ago, between 10 and 20,000 years ago, migrated from the north, slowly but surely, down into the Florida Peninsula, which geographically is like a funnel, right? You come down and there's nowhere to go eventually, and not back up. So that's how this area got colonized through the gene flow of Dade County/Pine, which established here. And before there was colonization of the peninsula, I'm talking modern times, the end of the 1800s, beginning of the 1900s, 120 years ago. The whole peninsula was basically one huge forest of slash pine, which also makes very good wood for construction and cabins and so forth, and that's how they got decimated, and now it's less than 1% of the county has these pine forests. And we happen to have one on campus, and we're very fond of it and trying to maintain it, okay? So that's an example of a climax community starting all from Pioneer, when this was all sand here, and some little tufts of grass established in the area thousands and thousands of years ago. Okay, so you're clear about migration in and out. It's at the level of the organism, at the level of the population of each species, colonizing a new area, moving from region to region, etc. Very dynamic flow. The next one is drift. Now, drift has to do with statistics, so it's a probabilistic model again, but it has to do with chance. And that's what we call a stochastic process. A stochastic process is a random event. It's a random chance. For example, flipping a coin. What are the chances? Because there are only two sides to the coin, right? The chances are that it will fall either heads or tails, so it's a 50-50 chance. That's a stochastic process. On average, a flip has 50-50 chance of head or tail. But each flip is either head or tail. You cannot get the same results on a single flip. So each flip is 100% either head or tail. So a flip, an individual flip, after the flip, it's deterministic. Before the flip, it's probabilistic. All right, so before the flip, you have 50-50 chance. But after the flip has occurred, then it determines it either one or the other. See what I'm saying? Okay, so it has to do with that random chance. And the random chance is that because these animals and plants and every living thing is in a dynamic state of flow in and out of regions and so forth, it's a chance occurrence who happens to be in the area at that particular time that the event happens. So here I'm just giving you an example of at least two populations. These flatfish, they're from the tang family, all right? And so these are blue tangs, which you see in general are going toward the right. And then you have these yellow and orange fish they're called wrasses, and they are generally moving toward the left. So there's flow in and out of this particular column of water. Here's a coral reef down here. All right, so it's along the coastal line more or less. You have two populations, one population of tangs moving in one direction, another population of wrasses moving in the other direction. But in general, they're in the same general environment, the same general vicinity. So this is definitely a community, right? There are other individuals there. There are other little populations that are less clear, but this is definitely a marine population, a marine community. All right. The fact that this particular individual tang is here at this particular time and it's not this other individual is by chance, all right? For example, you see the pattern of the face pattern on this particular tang? See the face pattern is different from the face pattern or the colors on this particular tang, right? It's more similar to this one. So the fact that this tang is here and this other tang is not here at this particular time is more or less by chance. Okay, it could have been this darker face tang that was here when the picture was taken, when the photograph. This is a photograph, by the way. All right, but the chance is that it was this tang and not this other tang. That's a stochastic process. That may have evolutionary effects if this lighter color represents a female and this darker color represents a male, then the chances are that this male will mate with this female as opposed to this other female that's further away. I'm just giving an example. All right. Let's look at drift, staying again with the probabilistic model so that you see where the chance comes in and how the drift introduces what is known as random bias or sampling error, and it's also known as the bottleneck effect. let's look at this series of three moments in the life of a beetle population, of a beetle community. Why am I calling this a beetle community? Let's look at stage 1. I'm saying that this is a beetle community. Why is this a beetle community?
Different
There are at
species.
least 2 populations of beetles here, if not 3. What are the 2 populations? We got green beetles and got brown beetles. That should be pretty obvious, right? Unless some people are colorblind. We got green beetles and we got brown beetles. Then in the brown beetles, we got large beetles and small beetles. We don't know if these small beetles are juveniles, all right? If they're juveniles, then they could belong to the same species. But if these small beetles, they can be large beetles and they can be small beetles, right? If these small beetles are also adults, then how many populations do we have here?
3.
Exactly. Beautiful. Thank you very much. All right. So one population has 3 beetles, the other population has 2 beetles, and the other population has 4 beetles, right?
Yeah.
Okay. Then this fellow comes along and he was not watching his step, and he stepped on 2 of the 3 green beetles by chance. It could have been the brown beetles, but he stepped on the green beetles. All right. So now, after the stepping and the catastrophic event, yes, for the beetle, it was only catastrophic for the green beetles, right?
Yeah.
So that after the event, now there's only 1 individual of the beetles, of the green beetles, but the other brown beetles are okay. Let's assume that the beetles reproduce sexually. This one beetle is on its way out, right? So 1 more generation and the green beetles are gone because of a catastrophic event that by chance stepped on the green and not on the brown. Excuse me for a moment. I'm going to break here for a moment. Need to pause for a moment. Yes. Excellent. Okay. Thank you. Back again. So you see how this chance event, which was dramatic and catastrophic for the green beetles, did not affect the brown beetles. Okay? But it was such by chance. It could have been that he stepped on the brown instead of the green and so forth. So that's the stochastic process. It's also called bottleneck effect for this reason. Let's assume that the marbles in this bottle are human beings, okay? And that the yellow marbles are people from Northern Europe, and that the blue marbles are people from Africa. In this original population, in this parental population, which can be somewhere in Southern Europe today, maybe the Italian peninsula, okay? There's an equal number of Europeans and Africans of the same species, right? So it's actually a single population here, okay? Now, some dramatic event, only the first few marbles go into a new region. Only the first few marbles go into a new region. And by chance, it happens, by chance, that there were more blue marbles than yellow marbles that came out from the tip, from the neck of the bottle. All right? So by chance, this group of people that were in the Italian peninsula migrated into the island of Sicily, all right? And by chance, more Europeans, okay. So here's the Italian peninsula, and as you know, it's in Southern Europe. Northern Europe is up here. and then the northern coast of Africa. In fact, Tunisia is very close to Italy. And then at the tip of the boot is this island called Sicily. And even though it's very close to Italy, there's actually a strait in between, the Strait of Messina, because Sicily is an island. All right. So back to our example here. This is Italian Peninsula. These are the Europeans and the Africans living together. And then this is the island of Sicily. All right. And guess what? The Africans knew how to swim, but the Europeans didn't know. Only these two Europeans knew how to swim. But more Africans did and made it across the Straits of Messina into Sicily. Generations went by, and it happens that I counted the marbles. There are 2 yellow, but there are 7 blue. All right. So it happened that it was 1 male Italian, 1 female Italian that got across, European, but then there were 3 males and 4 females of the African, whatever, but there was male and female who procreated more. The blue procreated more, the yellow procreated, and they procreated at the same rate, all right, but the starting number was fewer.
Yeah.
So the ending number, because of the geometric progression, the ending number is also fewer. So over several generations, the population in Sicily is much more African than the population in the mainland of Italy, okay? Just by chance, because the ones that got across were mostly African as opposed to European. Here's the stochastic process here. And that's why it's all called bottleneck. They're actually using a bottle to illustrate it, but the bottleneck is that only a few individuals go into the new region, into the pioneer region. And it is the genes of those individuals, it is a variety of those individuals to the exclusion of all the other individuals of the population that didn't make it across. So there was loss of variation, okay? But that loss of variation could be that the traits that are brought in by these new individuals have an advantage. They're more resistant. Maybe Sicily is a more dry area, and the individuals that went in had a greater chance of survival in dry climate. And so that will push that trait to be highlighted and could eventually lead into speciation if the difference becomes large enough so that if this population many generations later goes back into the mainland Italy, they will not recognize each other and they will not mate. And if they don't mate, then they no longer belong to the same species, right? Because one of the 2 characteristics is that they are able to mate, and the other one is to produce a fertile offspring. It could be subtle, but for them, it's real and drastic. I no longer recognize you as being a member of my same species. See how it works? All right. Another example. Okay, so what causes drift? Drift can be due to biotic factors or abiotic factors. We're on the final stretch here. What is going to be a biotic factor? Biotic for living, right? So a living factor. In other words, the agent of drift, the one that's causing the drift, is a living organism. In this case, who is the organism, who is the biotic factor? In the example of the photograph here, the cows grazing on grass, okay? You see that on the right, there's more grass than on the left, right? Because what's between the 2 patches? A fence. All right, so we have a population of cows here, and we have, 2 populations of grasses, one on the left side of the fence, the other one on the right side of the fence. What happens to the population on the left side of the fence? That grass is getting mowed down periodically by the cows, all right? And then they can get a little bit into the population on the right, just at the edge. So the periphery gets it. The periphery of grass gets it because the length of the neck of the cow can reach that far. This you would think, well, why doesn't the cow just push over that little fragile fence here? Most likely, this little innocent little white strip of fence has what? A little electrical charge on it, so that the cow gets a little zap. And therefore, the cow learns to stay away from the little white strip of plastic, all right? And that's why the cow just doesn't push over, because the weight of the cow is perfectly sufficient to push that fence over, all right? So the cow knows it can only go that far, but it will try its best to get into that new grass. So the edge gets it, the periphery gets it. This is a biotic factor because the cow is living, exerting pressure on this population, on the population of grass on the right. All right. Whereas, and it happens that this grass has the bad luck. This grass, of course, is on the left, and the grass on the right, this one has the bad luck of being on the edge. And that's the one that's getting the selective pressure. But it could also be due to abiotic factors. What's abiotic? The A negates, so non-biotic factor. What's a non-biotic factor? Well, a non-living factor. What's the non-living factor that is doing selection on this forest?
The fence.
A lightning strike.
Lightning.
Here's a forest at night with big trees. You see the big trees? And what's the bad luck of this tree? That it's the biggest of them all. And so it's the first thing that this light in a nanosecond is going to hit ground. It's looking for the tallest light pole, lightning rod. So this tree, by being the tallest, is a natural lightning rod. And this lightning, which is a discharge of friction, of static friction of the atmosphere gets charged, and it's going to find the fastest pathway, the closest pathway to ground, to discharge that spark, all right? It's a huge spark. It's like when at night it's cold and you move the sheets and you get little sparks. Those are little tiny lightnings, okay? When you do that at the level of the atmosphere, you get the dramatic effect of a lightning, which is one of the most, the strongest forces in nature. Thousands of bolts that come down through the atmosphere onto this tree, lights it up into a ball of fire in a split second, and then the tree will die, okay? Because thousands of volts go through it. So this biggest, tallest tree, which one can say that on average, being the biggest and tallest, was the strongest tree of the forest, just got zapped by an abiotic factor, namely the lightning. By chance, happened to be the tallest one. Okay, so on genetic drift, again, the factors exerting the drift can be biotic or abiotic. Again, the stochastic process also occurs at the level of the alleles, so you can also bring it down to the chromosomal level when we go back to recombination. And so this is kind of a recap of recombination, because here, as I mentioned, we have how many chromosomes? 2 chromosomes, 1 pair, 2 chromosomes, okay? They have the same size. One is blue, the other one is red. They have already duplicated, so this cell is getting ready to undergo mitosis. In this case, it could be meiosis. But the point is that on this leg here, there is the A gene, and there's a dominant version of it on the red chromosome, and there's a recessive version of the gene, indicated by capital A or lowercase a. So the dominant gene is called the dominant allele, and the recessive is called the recessive allele, because this is the same chromosome pair. This could be chromosome 1, 2, 3, 4, etc. for this ladybug. And what is the color red? Or any color of this bug? Is that the genotype or the phenotype? The genotype is the A gene. That's the genotype, is the A gene, which can either comes in two forms, dominant or recessive. The color is the expression. We're not seeing directly. It's not the gene that is literally red or cream. What we're looking at is proteins that are that color. So this is the phenotype. This square here is all the phenotypes of the A gene. All right? And there are essentially three expressions. If both are dominant, then it expresses red. If both are recessive, it expresses cream color. And if one is dominant and the other allele is recessive, it expresses yellowish, all right? Which is kind of a tone in between, right? So by chance, and this is it, so this particular chromosome pair here, with a dominant allele and a recessive allele, which one of all these bugs is represented by this particular set, capital A, lowercase a? This particular chromosome pair, this particular one, we have one dominant and one recessive allele, represents the middle bugs, the yellow bugs, see? Capital A, lowercase a, right? The middle bugs, all right? So when this pair expresses, with these two allele expresses, they will express in yellow. Not red, not cream, but yellow, okay? All right, by chance, these three red got hit by a rock. A rock fell on them, or a shoe, so these are gone. So now you got only one.
The three or two reds?
Two of the three.
Okay.
These two got hit by a falling rock, all right? Gone. And these two, they didn't find a mate. So they didn't reproduce either. So that's going to shift the ratio of the next generation. Certainly, if this is a total population of red, again, it's the same similar argument. They reproduce sexually. In the next generation, the reds are gone. The reds are gone. Okay, you can blame the rock. And this ratio is going to shift also. The cream color is going to stay the same ratio, all right? But the yellow color, the ratio, the number is going to drop exactly because two of the 9 did not find a mate. 2 of the 9. So only 7 of the 9 got to reproduce. Okay, so, and it was by chance. They didn't look in the right place. They wandered too far away. They got distracted by food, who knows, but they didn't find a mate. So you see the stochastic process, genetic drift. Summary of the whole thing. The 3 of the 4 evolutionary forces, mutation, which happens at the DNA level, either by DNA mutations or by meiosis, recombination in gamete formation, or by transposable elements, or migration and drift that happen at the population level, migration with the gene flow in and out of the population, or drift, also known as genetic drift, which will be that sampling error that because each population has a limited number of individuals, then the entire gene pool will be represented only by those individuals. Or those individuals will have some representation of the gene pool. By the entire gene pool of the species is larger than any one population, because no one population will have the full sample, the full gamut of the gene pool for that species. And then finally, I keep referring to selection that I'll cover on the next lecture. Drift occurs by chance, right? The stochastic process. And there's sampling error, right? Think of the 4 of us. We are all 4 adult men. So in principle, we could pass on our genes. We belong to the human species, but the human species is 7 billion human beings. On average, what's the ratio between men and women on average in the whole human population? Anybody know what's the ratio of men and women in the human population? You think more men, more women, or about the same?
2 to 1.
It's about the same. It's about 50%. And that's why when we do genetic engineering and when we do population control and things like that, they shift the population. Okay, when there's a selection, selecting out females, selecting out baby girls, that shifts the ratio of population. That could create huge social problems. Okay, but the ratio is about 50-50 more or less. So of the 7 billion, how many, we can estimate how many are men approximately?
3.5.
3.5 billion, right? 3.5 billion men, okay? We're 4. Now, we belong to the human species, but we don't have the fullness of the human genome that represents the 7.5 billion, right? We represent 4 of the of the 3 billion, but we don't have the fullness of that population, okay? No one group, no one sample is going to have the fullness of the genomic expression of the entire population. And so by chance, wherever we procreate and pass on our particular genes, yes, they're human, but they belong to these particular humans and not other humans somewhere else, okay? And so that's what I mean. There's always sampling error. Any particular sample, because the sample is smaller than the whole population, there's an error there. There's some others that are excluded from this particular sample, even though they belong to the same species. Okay, that's all I have for today. If there are any questions, comments, no? You've gotten enough? All right, we're gonna stop here then, and then I'll send you the PowerPoint and I'll send you the link for the recording. Probably by the end of today, you'll have the link of the recording, okay?
Great.
Okay.
Thank you,
so
Father.
You're welcome, you're welcome. Thank you, Eliza and Father Dayan. And again, summary is always due on Wednesday by midnight in principle. All right.
What's the next summary?
So, okay, okay, let me just stop the recording now. Stop recording.